Hi everyone. I'm a PhD student in Annalisa Massico's lab in Helmholtz, Munich, and today I'm introducing the method that we have been developing for quite some time now, the Graph Antiviral Target Explorer, a method able to predict disease genes in viral infections using graph message passing neural networks. So the challenge when Okay, so the challenge when when a new pandemic arises is that we don't have much information about it, and the computational methods can definitely help finding disease drivers. We have seen this, of course, with the SARS-CoV-2, but also that is COVID-19, but also with the monkeypox, Zika, and Ebola. The idea is that the interactions between the host organism and the viral proteins and the potential antiviral drug targets are quite complex and act at different scales, and understanding what are the molecular mechanisms driving it is quite complicated. And so we started integrating multiple omics specific for viral infections and also taken from related diseases. And the idea is that starting from those and the known host factors that are genes with proviral activity, we can find other potential host factors that can also be potential antiviral drugs. And the idea is to develop something that can generalize to less studied or maybe more dangerous viruses. So the data that was available for us comes from our collaborators. So we have plenty of data about SARS-CoV-2 that maybe I can. Okay, okay. So here we see 4 different omics type for four different omics type for the SARS-CoV-2. So we have transcriptomics taken at 3 different time points. And then the proteome abundance taken and fold change of it taken at also 3 different time points. And then also the host viral interactome, that is basically like the interactions between the 24 viral proteins of SARS-CoV-2 and the host proteins. And then the host effectome, that is another interesting signal that measures the protein abundance given the overexpression of each one of the 24 viral proteins in SARS-CoV-2. Then we have 190 host factors that are our positives and basically 20,000 other potential negatives. For monkeypox, that is a bit less studied, we also have transcriptomics and proteomics, but in this case, the data comes from primary cells. So even if the omics are less, it is still quite a relevant signal. But we have no virus-specific host factor, and we take those from the vaccinia virus. And and I will explain how. So we started implementing, working with graph message passing neural networks. And the idea was to build a network that is using protein-protein interactions and based only on the host organism, so without including the viral proteins at the moment. And each node is a gene and can be either labeled as influential, like important, a host factor that plays a role as a pro-viral activity, or not influential. This means that we think that that it might be not important, so potential negatives. And then we have all the unlabeled nodes, those that we have doubt about it. And so we want to predict if they are host factors or not. So the idea is to build use multi-omics to describe each protein in the network, and then stacking layer after layer of this graph message passing neural network. So we want to enrich the representation of each protein and come up with a better representation of it. Okay, so We started analyzing those 190 host factors that come from a review paper from Baggen of 2021 that is collecting 36, the results from 36 different studies on SARS-CoV-2. And they also report manually created, functionally validated host factors that are these 190, and a lot of others that are, let's say, less trustworthy. And then we also have some drug targets and that come from another publication and are reported in this therapeutic target database, still specific for SARS-CoV-2. And the idea was to see, okay, let's see how are they placed in the network. And so we see here with the stars, we see the drug targets, and with the blue dots, we see the functionally validated host factors. And what we can observe is that they only partially overlap and mostly form clusters in different places. So those that are good host factors, also experimentally validated, are not very often good antiviral drug targets. So we started thinking about how to bring these representations a bit closer. And because this would be definitely challenging for a message passing neural network that is aggregating information from neighbors. And so we decided to go for a pre-training task on a general purpose task. And the task for pre-training becomes classifying genes that are related to any kind of disease, and simultaneously also genes that are reported as potential drug targets. So here now I'm describing like the three different inputs that we are using for our pre-training step. So we have this positional encoding that is done on the PPI network. And so here we use a random walk with the restart using the weights given by the different strength of the connections. And basically here we see like how likely it is for each protein in the graph to be visited by a random walk restarting from all the other proteins. So we get a representation that is, in this case, 18,560 long. Then we have, we also decided to, so okay, the positional encoding, the idea was that since drug targets and host factors have different positions in the graph, this might help the learning process to bring the representation closer. Then we have the protein sequence embedding from the pre-trained ESM2 model. And the idea here is to use a simple pre-trained ESM2 model, then do an average pooling, and use these also afterwards for message passing, because this should help retrieving the interactions that might happen in protein complexes, and so help the classification. And then we also used some functional embeddings taken from gene ontology semantic similarity matrix that is obtained using the GeoSemSim-R package. So this is how the pre-training looks like. So here we see, let's see if I can use the cursor. Okay, so here we see the positional encoding that we have seen before, that is then reduced in dimensionality using a PCA, and then with a linear layer plus activation is brought to a specific dimension. Then the same happens, like, no, not really the same, but similarly for the large language model protein embeddings, we use, in this case, the message passing to allow neighbors to contribute. And then we come up with another representation of the same dimension. And then we have the gene ontology functional embedding that have a similar process to the one of the positional encoding. The task, as I said, is to classify predicted, so genes that are related to any kind of disease, like from this part here, and potential drug targets. And we take this data. So as a network, we use a string. and the training positives are taken from the comparative toxicogenomic database, using only those that are manually curated as as real connections and not the inferred ones. And then we use drug targets taken from drug bank. Then the fine-tuning to a specific virus. So in this case, this must be virus-specific. And so here we can integrate together different omics with the different input dimensions. And the idea is to stack them together, bring them to a same dimension, sum them up. And even if this maybe sounds a bit simple, it demonstrated to be quite effective. And then we use the message passing neural networks here, and we aggregate them to the final representation that is used in principle for classification. So now we can put pre-training and fine-tuning together. So here we have the pre-training block that is done on this general purpose task. And then after we learned how to classify genes related to any kind of disease and drug targets, we isolate the head that was predicting disease-related genes, and then we plug in the fine-tuning model with the omics data or any other actually kind of data that can be specific for the virus. Then we remove the part that was doing the classification of the drug targets, and we fine-tune everything to the specific virus, trying to predict what we know about this virus. So here now I'm presenting some of the results and some of the main things that we understood during this process. So what we can see here is that so when we use graph message passing neural network like a GCN or the principal neighborhood aggregation, we get the best results. Then if we use just a simple multilayer perceptron, the results are already quite good. And in this case, also positional encoding is included. If we remove the positional encoding, we have another drop. Then it's interesting to see, like visualizing drug targets, we can see that the drug targets are predicted better if we use message passing neural networks. While the difference is not so evident for the functionally validated host factors. This is the time. Okay. Then we did an ablation study, like seeing like which are the most important inputs. So it's interesting to notice for SARS-CoV-2 that is the one that has the best performance, also because the positives that we are using are quite good. So here we see that protein embedding, like if we remove the protein embedding, we have a drop, but especially if we remove positional encoding. And also the omics are helping a lot. Instead, for monkeypox, like the biggest drop is when removing pre-training, and this, in our opinion, is due to the fact that the information that we have on monkeypox is not much, and pre-training is actually helping a lot that task, while for SARS-CoV-2, maybe a bit less. Then so this is also other evaluations. So here we can see the predicted probability of all genes versus those that are potential host factors, always from this Bug and et al. Publication. So not very reliable host factors. And then the drug targets from the other database that I was showing before. And we can see that for SARS-CoV-2, also for monkeypox, taking drug targets from another, actually extracting the drug targets from the drugs found by our collaborators, we see that those are predicted quite well. And the precision at K for the drug targets is quite good. This means that like taking the first 100 predictions, 6 out of 100 are drug targets that from the independent validation set. and in this case 7. So considering that the set is around 20,000, it's quite a good result. Then we benchmarked against sequence-based methods specific for SARS-CoV-2. So we have this LSTM-based and another transformer-based method. And we see that the precision at K and also EUROC and AUPRC under all metrics, gate is performing the best. And and actually we can also see that the LSTM-based method is performing better than the transformer one, at least from our benchmark. Then I did an explanation analysis that is, so basically checking, applying the GNN explainer, finding the most important features and subnetworks. And then I wanted to check what happens if I mask like the important features and the important features from the source node here on the left, and the source node that is the one that is predicted and that we are trying to explain, and from the neighboring nodes that are the most influential ones. So from the neighboring nodes, we see that positional encoding and the gene ontology semantic similarity are not contributing because they are not part of the message passing. So this is normal. And but we see from the source node that positional encoding and also the omics are contributing the most. And and also from the neighboring nodes, we see that by removing them, the probability drop is also quite high. And then by doing a zoom into the different omics, here I forgot to say that this is from SARS-CoV-2. And here we see that the interactome is the most important. And this is also kind of expected from the way the positive set is created, that is using heavily the interactome. Then we can also analyze the neighbors. And here we see from the top 100 predictions that are in yellow. We see also that like the most important neighbors that are bringing this classification that are labeled in blue. They are also kind of close to each other, and in some cases slightly more far away. And we are also able to see for each specific prediction the subnetwork and see like the performance and see which features are contributing the most. And we have this case of the beta-catenin in which we see, for example, that FRAT1 and FRAT2 we see the contribution of the protein embedding part from the language model, and this makes sense because they are physically interacting with it. And then we also have the connection with this other one, this SMARCA4, that is a host factor. Then doing okay, I cut it short, okay. The gene set enrichment analysis also shows that the same functions that were enriched for the positives are also enriched for our predictions. And then, okay, so I guess I have to cut it short. So our method is quite flexible, doesn't need much data to be trained since there is the pre-training part. And we can also, since the interactome is very important, we can also work with the viruses that are dangerous and for which we just need to overexpress the proteins. And I have to thank the people of my group that helped of Marsico Lab and also of the Innate Lab that provided the experimental data. And thank you.
Thank you, Samuel, for bringing us to microbes. We will take the questions offline since we are out of time, but